floor is now open for comments, additions, alternative ways of looking at the science of aging. Please denote your interest in making a comment by picking the hand raise function on your reactions. Section Panda. You have to unmute, Satchin.
Yeah, I unmuted, and hopefully you can hear me now.
Yeah.
Yeah. Thank you, Eric, for that fantastic talk. I'd like to add another aspect to aging, which we work on, that is circadian rhythm. Circadian rhythm, I believe, is an emerging area that has untapped potential to understand fundamentals of metabolism, behavior, physiology that affect the aging process, and can extend healthy longevity by impacting not only at personal level, but it has scope to even formulate new public policy that will impact longevity at community levels. So the best connection between circadian rhythm or disease have been studied in many different levels, from animal models to humans. But I guess the best connection we can make is how shift workers, people who work in day and night shifts, how they're impacted by circadian rhythm disruption, and they are at a very high risk for age-related chronic disease. And in fact, every time you hear a story about a person who lived up to the age of 100 years or longer, you'll rarely find a firefighter or a nurse or a shift worker or a physician living to the age of 100 years. So that has given the field a lot of work to do. And I think in the last few years, the emerging questions from circadian rhythm research have been around this one big breakthrough, that is nearly every gene in primate genome or animal genome has diurnal rhythm in tissue-specific manner. And these rhythms seem to optimize metabolism, behavior, and physiology, and protect against or increase internal capacity or resilience. So that has led to three implications with potential impact on longevity. First is how to nurture healthy circadian rhythm to sustain these rhythms, and then how to optimally time and the timing of medications and medical procedures to increase treatment efficacy and reduce adverse side effects of a large number of drugs and procedures. And then the third one is more relatively new, whether drugs that target circadian clock components can be effective in treating age-related disease. At the same time, it also raises additional questions, what we already know about aging pathways. For example, Eric, you alluded to subtwin, mTOR. Then the question is how the circadian mechanisms intersect with these known pathways of aging. So I'll pause there, and then maybe I'll participate in discussions again.
Okay, thank you, Satchin. Before I ask anybody else to comment, there are a number of people who are watching the live stream. They're not the discussion group, but you can send your questions in or comments to healthylongevity@nas.edu. And we will try to get your questions addressed. I did have one comment, Satchin, which is there an understanding at some molecular level of the impact of circadian rhythm on aging?
Yeah, so there are quite a few studies done in animal models where genetic disruption of circadian rhythm or chronic disruption of circadian rhythm by imposing a shift work-like lifestyle on animals can reduce their lifespan. And these studies are done in completely aseptic condition, as in all vivariums. But if we do these experiments in real world situation, I guess the reduction in lifespan may be even more, because when we expose animals, healthy animals with circadian rhythm disruption to non-lethal dose of bacteria and other pathogens, then these disrupted mice have more severe symptoms, and even they can die from otherwise sublethal dose of pathogens. So in that way, chronic circadian rhythm disruption can compromise longevity by two different ways. One is by the intrinsic molecular pathways by which it supports health, which we do not completely understand. And second, by interfering with the immune response in a way that the animal or humans may be mounting a hyperimmune reaction to a pathogen.
Okay. Thank you very much, Sachin. So the next question by Nir Barzilai.
Thank you. So first of all, Victor has not been lazy. Last time I saw him was about 2 years ago, trying to avoid COVID in Singapore, right? That's where I saw you running last. And Eric, not only that was a good summary, but it also shows, I mean, for all of us, how united we are. There's no controversy on what Eric said. We're totally believing in it. Each one of us has the example. So I want to suggest to frame the discussion of for the National Academy of Medicine is eventually we want to treat patients. Okay, let's think about that. We want to prevent age-related disease in patients. And there are two things that happen in the field that have allowed that to progress already. And when I say progress, biotechs, there's almost $1 billion investment in biotechs in the field of aging-longevity. And what allowed it are two things. On one hand, what Eric, I don't know if you said the word, but the pillars of aging or the hallmarks of aging, really. Those 9 or Satchin included now a new one, but those things that we know are going wrong with aging, and if you fix it, you get an extension of health span and lifespan, and miraculously also they affect each other. In fact, you don't have to correct all of them in order to get pretty much increase in health span. So this is one thing that allowed investor to go and said, you know, I want to target proteostasis on autophagy, or I want to target metabolism or something else. That's an important thing that we have to keep in mind. We have to develop those hallmarks and get more of them and get them in hierarchy and a whole other discussion. The second thing that is really important, has been very frustrating, is to get the regulatory system like the FDA to agree that aging can be a target, that we can target aging, and in this sense, we can increase health span significantly. And this is, and the TAME trial that's supposed to do that and hopefully will be launched post-COVID soon, is a trial where we're trying to do it because without that, we're kind of stuck, where there's no indication for us to go unless we actually show that we're going to be successful. So I just wanted to point up to those two things and to suggest that that's a major frame for us, because those are the things that will help us get to the patients and prevent aging and increase health span. Thank you.
One of the challenges of aging as an endpoint for the FDA is really how long will it take to reach an endpoint that's measurable. And within the time frame for clinical trials, it becomes very complex. So if you have to wait 4 or 5 years before you get a result that is registerable, the kind of investment that's required for that is, I would say, quite challenging in the current environment. So I,
yeah,
I don't think the FDA is resistant to it. It's just that companies don't ask for it.
Well, companies don't ask for that because the FDA is not aligned for that, or you know, the FDA is expecting TAME to come and show them that it can be done. I just want to make 2 points. I think with biomarkers, we'll be able to find sooner. We don't have to do a 5-year study to fail, okay, if we have the right biomarkers. So I think it will be very helpful. The second thing, the TAME study, what people are surprised, we're trying to prevent several age-related diseases like cardiovascular and cognitive and cancer and mortality. And people are saying, are you crazy? That's a lot of diseases. But we need less people because we don't care which disease we're going to prevent. Okay, we need only 3,000 people because it doesn't matter what disease they have. We're going to shift the next disease away. And so it's going to be less expensive, at least, to do trials like that.
Okay, Felipe?
Yeah, I'd like to just thank Eric for a very good summary. I'd like to come back to his last point about what are we gonna do with 20 years of healthy life? Because actually, as Eric said, the system right now is focused on taking care of the sick people. That's all that the system does. And if we are successful in what we're trying to do, we're going to find ourselves with a whole bunch of people who are 90 and in very good shape and very good health, ready to work, ready to participate in society, ready to participate in life, and they're not given the opportunity. I think that's something that we as biologists are not often thinking about, but it's critical that we consider that aspect of what we're doing, because as Eric and I think all of us in here are convinced that yes, we will arrive at what we're trying to do. And we will arrive at being a preventative system where we're not really going to be taking care of, as Nir said, of patients. No, we're going to be taking care of healthy people and making them remain healthy because it's a preventative system. So that's, I just want to put that into the table because I think it's an important point.
Thank you, Felipe. Andrew?
Hi, yes, and thanks again, Eric. That was a really great summary and provoking in many parts, I think, to guide this discussion. One of the things I wanted to touch on was the biomarker. And maybe there's an FDA issue here of getting a trial through success and the length of time a trial would be. And I guess one of the things I'm sort of questioning is what are the biomarkers? And without a good biomarker, where do we go? If we were thinking about the reference in the hallmarks, right? It's by and large animal studies, or if we think of diabetes and sort of inflammation, are they the biomarkers of aging or are they the biomarkers of being unhealthy? Now, I think solving being unhealthy is a big thing and a lot of people benefit, but I wonder if these hallmarks are a feature of health span or are they a feature of disease, and can we uncouple them? You know, we tend to think of aging as a monolithic problem, but if we think cancer is cancer, or if you try to treat COVID with Hep C antivirals, right, we have a big problem. And so I just wonder, when we think about mouse studies, going to human, are we going to be able to find the right biomarkers? And I think that'll be the gateway to facilitate or nudge the FDA, if you like, to maybe reclassifying aging as a disease. If that is indeed important, I think there are a lot of drugs out there now that are in trials for different things, cardiovascular risk, which, for example, which probably are changing components of health span. So just a few thoughts there.
Thank you, Andrew. There is a question from a listener that I think is relevant here. The primary metric for aging interventions in animal studies has been mortality. So where are we with models in which health span is the primary outcome for interventions in animal models? This is from Jeff Halter from the University of Michigan.
maybe I'll just, as I'm on, I can just respond briefly. There are actually a number of scales and sets of tools that are being used in the field that have been validated as surrogate markers for mortality and actually are highly predictive of mortality. I won't go through them. There are many of them. One thing that I really would like to sort of further amplify is the point that was made by Andrew, is the whole idea of biomarkers. I think you cannot treat something that you cannot measure. And in this respect, I think what's happening with the field of biomarkers of aging, I think is probably one of the place that with the funnel by which we're going to have to go through to really implement some of these discoveries. Of course, no one is considering 30-year clinical trials in humans, and the markers right now, the biomarkers of aging are proliferating. We have Horvath clock, we have proteomics clock, Tony Whisker-Ray, we have transcriptomics clock, we have IH from David Furman at the back. We have the SASPOM, which measures all the secreted protein from senescent cells. And we have all kinds of other interesting biomarkers, such as, for example, facial recognition or markers for skin. There's a lot of activity in the field, and my prediction is within the next 5 years, some of those are going to emerge as the most important ones to follow. Just a couple of points.
Thank you. Tom?
Great. Thank you, Tashi. And again, thanks, Eric, for a great overview. I just wanted to highlight something I think has been touched on by several people, and that is a shift that will happen is not only kind of removing of these silos of neurologic disease and cancer and so forth, but in a way, a demedicalization of what we're talking about, because we're still talking about FDA and patients, and it really is a profound shift, I think, to consider all of this in terms of much more public health, as opposed to what doctors do in terms of treating patients, as opposed to what we do. And Eric, you alluded to, of course, the importance of thinking about interventions that are not just drugs, but lifestyle. And I know this data is somewhat controversial, but when people have looked at yeast or mice and looked at different genotypes, caloric restriction can shorten lifespan as easily as it can lengthen lifespan. So I think that really highlights the importance of lifestyle interventions, maybe first and foremost, as a direction forward, even as we develop drugs that will treat diseases and then will maybe treat aging. I think there's no doubt that really lifestyle interventions are going to be far more profound and really remove it from the idea of medical doctors taking care of these problems as opposed to public health.
Victor?
I want to be provocative. I think everything you say, I agree. So it's not a disagreement. However, as we think about technologies that's going to be able to, in fact, achieve regeneration, longevity, you name it, there will be a time where we have to address the issue of ethics, equity, and societal implications. Let me take one extreme example. Partial reprogramming. Eric, I'm sorry you didn't talk about this. It's such a breakthrough technology. In partial reprogramming, you can reset your cell to a clock to 0 and start again. Now imagine that this technology is useful in human. I would like to take it at age 45. Why 60? And when you start thinking about all these great technology that can be used earlier and earlier, one begin to ask the question, are we doing enhancement or are we doing something else? In the traditional way of thinking in medicine, we still think about disease treatment. Yes, you can do all these things in order to treat disease. But now as we begin to think about truly changing a health span, particularly in the area of partial reprogramming others, one could argue, number one, that's enhancement. In addition to that, the whole issue, aside from ethics of equity, Tachi and I just both tell everyone think about equity. So who would have access to this? And are we going to create a technology divide? And in fact, affordability, you name it. So I'd like to at least be provocative and ask that question for us to say, maybe we don't have an answer. At least we need to acknowledge that as we get better and better, we will be pushing in the envelope of societal issues. Thank you. Eric, you like that?
Yes. Do you want me to give it a try?
Sure.
My response to this question, there are two questions that are sort of valuable and important for our field to address. One is the question is overpopulation, decreased natality, and the societal crisis that if you take this population change to the extreme, are we going to contribute to more overpopulation, to more ecological problems? So that's one. The second one that you mentioned is equally important, which is equity. And right now, I can tell you the strongest predictor for your lifespan is your zip code, and along with your VO2 max. Why is that? Is because of a whole series of decisions. Now we have, as we all know, a society that is now splitting across economic lines, and I do not think that the aging field is contributing to this. It is a manifestation of a society that is splitting across economic line. And my argument has been for people that if you think about the whole way we develop drug through FDA approval and reimbursement from pharmaceutical companies, it's a process that actually has been given preferential access to the people who have medical care, who have access to drugs, who have access to doctors and so on. But eventually, all of humanity benefits from this. And the best example of this is actually the TAME trial, because the TAME trial is using a drug that costs 20 cents a pill, and that is basically being developed by the traditional FDA-approved process. Oh. So you can imagine the same type of approach for many of the medicines that we will develop as part of the aging field. They will be first restricted to people who can actually afford them using the FDA approval and insurance reimbursement. But in the long term, humanity will benefit from it. I think that's usually how I respond to these questions.
Yeah, no, I agree with you if, in fact, as simple as rapamycin, a drug. But we are talking about technology, genome editing, partial reprogramming, which will be very different. And those technologies, when applied, could raise a lot of different issues. You can apply them very early. You can increase your muscular strength. You can increase your, overall, the whole application. So I think it's not only an issue of access, affordability. It also crosses some lines that, at least today, what our ethical lines are. I only raise that to make a complete conversation. I don't expect an answer to this.
Right. So I want to focus a little bit of the discussion on new directions in science. Understand the perspective, the perspective that we have to address aging as an indication, then we can address a whole bunch of other problems. But there are advances in, say, understanding of the impact of DNA repair or senescence factors, or there are in solutions such as alternative ways to induce cells to regenerate or stem cell transplants, all these things. What are those areas of emerging science that are going to make a difference for the future? I do want to spend a little time focusing on that and not the overall gestalt of aging, which we all understand. Anna Maria, I believe you had your hand raised for a while. I'm sorry, I couldn't see your hand raised because it's against the yellow background of your file folder of bins up there.
I know, I realize I don't have the right background for this. I apologize. No, thank you for noticing. No, thank you for the opportunity, and also thank you for the great discussion so far. So my comment touches a little on what you were saying of emerging areas, with the idea that I don't think we are done with the areas that we are already involved analyzing. And something that has been discussed has been this idea of the hallmarks of aging, the process that more or less each of us has been characterizing. But Andrew touched upon the biomarkers and kind of mentioned, well, all these studies have been done in mice, and I agree, these are experimental models. But once I still think we are moving into readouts of health span, even with these models. I mean, you have beautiful work from Rafael de Cabo doing frailty in these animals the same way that we do in the clinic in geriatrics. So you get functional readouts, not of disease, but functionality of these animals, of course, adapted to the mice. So I think we are getting there with this idea of how much we can translate. And then many of these hallmarks of aging, to call them somehow, many of these cellular and molecular processes, many of us, even in this group, are interested in testing them in humans. So it's not going to be, for example, Nir mentioned proteostasis. I cannot be in a talk without mentioning autophagy at least once. So of course, we've been looking in the centenarians and trying to see if this parameter is also altered. So I think the efforts are there, but you are completely right about the need of good readouts. And I will make the push not for a single biomarker, but I think there are going to be signatures and different combinations. And you can have this successful aging through particular combinations, and it's not going to be unique. And I think it's our job and something that we have to really think moving forward and keeping patients and population in general in mind. How are we going to study that? How much can we benefit of populational studies like one of us that are going to get all that information, some of them very related to some of these cellular processes of aging, how much we can do to differentiate combinations and signatures that are going to predict healthy lifespan and healthy aging or not. So I think those are the things that is still pending. We don't know enough about that, but we might have an opportunity with these large studies coming.
Thank you. Joshua?
Hi, thank you so much. It's such a pleasure to be on this panel, and I want to thank the organizers for including me, and thank Eric for his amazing overview. I wanted to echo what Nir said, which is, I think amongst a group like us, it's not controversial, but we have a lot of challenges with regulators. I wanted to just mention an approach that our group has taken, which is to use mesenchymal stem cell infusions to extend health span. And I wanted to just mention the endpoint we've used in our trials. We actually have just completed a large trial that was funded by the National Institutes of Aging, and the results will be out just in a few months. The endpoint we chose was, I have to thank Linda for her pioneering work in this area, but we chose frailty as the endpoint of the trial. We enrolled individuals who were frail, and frailty is something that's measurable and quantifiable through a number of metrics. It's well described. And the FDA was responsive to that. So there's a potential here that trials can be designed that are smaller, that give answers regarding functional status in humans that might be more laser focused on the issue of health span that we're trying to treat. The one last thing I would say is that at the end of the day, trials and evidence are the crucial sort of valley of death that however we conceptualize this, we all have to traverse that valley of death, obtain the funding, do the trial with the right endpoint, and then hope that the FDA will be supportive of that concept and allow us to treat patients. Because I think one thing that I think there is a consensus on this panel is that aging and health span are treatable, and I would dare say must be treated. I think that's the ultimate conclusion of this panel, is that we really must focus on this challenge and That's basically my comment.
Thank you, Joshua. I couldn't agree with you more that evidence is needed, and evidence that is an endpoint that can be measured and acceptable, because the experience in almost every other therapeutic area is that animal models don't always translate to humans. Much of the aging evidence is based upon animal studies, and animal studies, unfortunately, are not always predictable, often not predictable. Certainly in certain areas like cancer, absolutely not predictable. So human studies, what kind of human studies can be done, under what conditions, and what kind of interventions can we think about, and how do we measure that? That seems to me a pretty important need in this area. Linda, you had your hand raised?
I'm muted. Hi, everybody. Thank you for this really exciting set of presentations and discussion. I just, from the point of view of the National Academy of Medicine Commission, I wanted to just reflect on something and then ask a question. The reflection is that a number of points are being raised about, as the science moves forward, what the implications are that we simultaneously need to prepare for. Whether it's the concerns that Victor raised about equity, whether it's the issues that Eric and others have suggested in terms of how we elevate the floor and the ceiling of health for everyone through perhaps a different kind of public health system. And I think we need to link the science deeply to the implications of how we will deliver it and carry it out. At this moment in time, when the public health system really needs to be reinvested in, given all the evidence of the consequences in COVID of 60 years of disinvestment, certainly in the United States, it strikes me that this is a very important time for our field to be making the points of how the reinvestment could go towards the creation of health span with lifespan. The question I have actually is going back to Eric's talk. Eric, you made a point about aging and the role of resilience in aging. And I was wondering if people could talk about where you think that science is and where it could go.
Eric, did you want to comment?
Much of what many of us actually are occupied with is understanding what these repair and protection mechanisms are. And, you know, a classical example of this taken to its extreme are patients who have defect in DNA repair, which show, you know, dramatic acceleration of aging. And much of, you know, the pathways that have been elucidated actually are dealing with understanding what is the nature of these repair pathways, what are the types of injuries that they mitigate, can we activate them, can we, even in the absence of the injury, there's a whole field of what I find fascinating, the whole field of hormesis, this idea that many of the interventions that we know actually increase lifespan and health span are triggers that induce some degree of stress. We're thinking about fasting, thinking about sauna, and thermal shock and so on. So there's I mean, this is essentially what many people in the field are preoccupied with. I see Nir wanting to comment as well.
Yes, there are studies that are being done across the United States now. NIH supported, I think Jim Kirkland is the PI of that, where you're taking several gerotherapeutics, let's call them gerotherapeutics, and giving them to people, for example, before elective surgery, or when they're going through an event that usually, because of lack of resiliency, they are either at risk of not recovering or recovering slowly. So in fact, those are kind of the clinical trials that we're populating now everywhere to check how those gerotherapeutic are increasing resiliency. As for equity, I just wanted to mention something that I think it's important for us to know. We're talking about aging, but you know, people who are surviving cancer, they're aging very rapidly, in particular the kids. They need help. And people who have HIV are getting diseases 10 years before their cohort. And people who are disabled are also aging very rapidly. And maybe the two opposite examples are the poor people. Look, the poor people also don't exercise because they cannot afford gym or gym equipment, and they eat very badly. And maybe a drug like metformin is going to be actually helpful for them. I mean, we have to think how to be creative with them. We cannot tell them to exercise and expect them to exercise or to diet. And on the other hand, if we're going to go to Mars, and if this is really something we want to do as the world, as a society, we're not going to go there before before we solve the aging, because by the time we get there, we'll be sick and we'll never come back. So there's many reasons for us to frame equity and everything else, and it's not only about aging, it's really important for so many other people.
Benjamin.
Thank you. This has been a great discussion. Along the lines of new science that may impinge on this, I wanted to bring in recent findings from the cancer research world. We often think about cancer as a disease you get when you're older, but the origins of cancer clearly happen at a much younger age, and we understand pre-malignant lesions at a level that we never, just in the last few years, that we didn't previously. With age, we really become coated in all of our solid organs with somatically mutated clones bearing true tumor suppressor and oncogene mutations in the skin, esophagus, liver, lungs, and they are widespread, and they are tiny microscopic clones, but they are all true pre-malignant lesions. And they are so widespread that they likely affect the function of those organs and the aging process. The blood is a special example because the clones aren't spatially constrained and they become huge. And that's a process we call clonal hematopoiesis. And that has a particular impact on aging because the mutated blood cells elaborate the inflammatory cytokines and the methylation signatures that have already been associated with aging and may be a cause in some cases of that aging process. So I think that's both an aspect of aging to understand, but it's also an opportunity. We could potentially treat clonal hematopoiesis or its consequences. We could develop assays to understand clonality because we don't have them in the solid organs, and we could develop approaches that would shrink the size of pre-malignant lesions and therefore decrease the likelihood of them progressing to full-blown malignancy. But that's potentially a new frontier for aging.
Ben, how is this related to some of the evidence about DNA repair mechanisms, that altered DNA repair mechanisms account for a lot of the aging process?
It's a great question. So as was mentioned, individuals who have genetic predisposition to cancer often have mutations that germline mutations that lead to decreased DNA damage repair and increased susceptibility to cancer. But the vast majority of the mutations that happen in otherwise healthy individuals are C to T mutations that occur with spontaneous deamination of methylated cytosines, which is just a chemical reaction that happens over time and is not related to any germline predisposition. So a lot of this is going to be just and more of an inexorable process that happens with aging chemically.
Tom?
Thanks, Taiji. So first of all, I would like to thank Eric for the great overview. And actually, I would like to echo Comrando's point on the lifestyle intervention. And actually one thing that I would like to point out is I think we have not talked about is exercise. I mean, Eric pointed out that one of the best prediction of life expectancy is the zip code in the US, right? So and actually in Hong Kong, our average life expectancy is very high, and I do think it has something to do with people walking around. For example, old individuals actually have to go to the wet market and walk for a long time. Going back and forth, even though they're 80 or 90 years old. So I believe, just going back to Tachi's, refocusing earlier on the emerging science and pathways. I think one of the areas that we should take a look more is the molecular pathways that considering about the regulation of these pathways about exercise, and that's the point that I would like to raise. Thanks.
Okay, Masayo.
Yes, thank you very much for letting me participate in this great discussion. And about the exercise he mentioned, actually it is known that the running will increase the regeneration of intrinsic neural stem cells in the hippocampus. So such kind of approach is very useful. But let me talk about the regenerative medicine that I'm in. And we have started to do the regenerative medicine cell therapy for the retina, the neural retina, that is the central part of the central nervous system. And now we are treating very severe case, but I'm sure the target will be more and more earlier stage. And finally, maybe the replacement of the senescence cells will be achieved. So in that case, the senescence or daily life will be maintained. And during the development, I feel some issues about the developing regenerative medicine, because in the case of replacement of the senescence cells, the target will be enormous people, not the disease, but all the people will be the target. So that about the equity, the cost is very important. And during the development, I feel in such case, we need a new regulatory system, completely new one. And the regulatory system now is for the small molecule and the end products. But also the end point will be changed, because the disease, about the disease, end point is usually the improvement. But the treating the senescence, the end point will be maintenance. So it will take a longer clinical trial, and not many companies try to do it of the 5 or 6, 7 years clinical trials. So the regulatory system should be changed completely. And also the long clinical trial cost very much. And also for example, the cell therapy, the license will be very complicated. We need many, many licenses. And nowadays the licenses are like money game, and that will most of the cost of the treatment. So we should change the rules or regulation, I think. And then the regenerative medicine or replacement of the cells might be a very good approach of the senescence. That's my issue.
Thank you very much, Marcel. Now there are several speakers that have raised their hand. I have to defer to my boss here, Victor Dzau. Had a question, so
Oh,
Victor, you have.
I was just following Tom, Ben, and Masayo and others about New Frontiers. I mentioned it, and I didn't know whether that had any traction, so I want to say it again. Partial reprogramming, to me, is one of the most important discoveries in the aging field. And as you know, Del Monte showed that using Yamanaka factors, transiently expressed in cells, and in this case, mice, progeny mice, have greatly enhanced and reduced aging and hence life expectancy. And it's an epigenetic mechanism whereby they reset the clock, the cell clock. I think this is a very important observation. It's theoretically, if it's done right, it could be a breakthrough and easier than, say, cell regeneration and many other technologies. I thought this at least should be included in our discussion. I'd love to hear maybe Chris Walsh or Tom Randall and other people's reaction to this.
Yeah. Reactions to partial reprogramming as a therapeutic intervention?
Yes. So
I'm raising that to the floor. Tom?
Yeah, I mean, so Victor, thank you for bringing it up. Again, I agree that's this is really an advancing field very, very rapidly and very, very new. And it has a lot of implications, I think, as you say, because it is really, in a way, resetting and potentially enhancing the functions of cells in the body. The challenges, so I completely agree that this is an interesting, exciting area. The challenge is, the question of safety, will this be possible to reprogram aging without reprogramming cellular identity? And I think this is where the field is. We work on this too, because obviously the risk, I mean, the field is at a very fine line between reprogramming aging and causing cancer, essentially at that balance. And I think that's where this field will evolve, either to a safety area where it's possible to reprogram cell age and not cell identity, or it will be too narrow a therapeutic window. I mean, at this point, it's a very, very narrow therapeutic window. And of course, it's a genetic therapy at this point. But there's no doubt, and we had worked on this again, this dates back from work we did on parabiosis, which has the same kind of flavor of resetting the clock. And we've been very interested in this area. And the question really is, can we uncouple that reprogramming of age and reprogramming of cellular identity, like with iPS cells?
Yeah.
So I think, yeah.
Yeah, Tom, of course, this is transient, expression, not long-term. As you know, the studies are done as a way to turn on the clock short, and Yamanaka factors for a short period of time.
It's true, but even then, it requires very, very fine tuning in terms of dose and timing to not go too far. So that's the promise, and I just wanted to raise that issue of it's a very exciting area, but there's a big safety issue there too.
Thank you.
One question that's been raised from viewers, the gender context for aging and interventions in aging and novel mechanisms that might both cause and address the aging process. Any comments? Chris Walsh?
Felipe.
Yeah, hi, thanks very much. This has really been a wonderful discussion. Just in terms of novel mechanisms and reprogramming and sort of new frontiers, I guess I'll just highlight some emerging work that's starting to study the genome of individual cells. And so it's been familiar for a long time that in cancer we see, and work like Ben's and so forth, that dividing cells accumulate mutations as they divide. But I think there was a widespread assumption that non-cycling cells like cardiomyocytes or neurons in the brain were protected from the sort of genomic damage that has to happen when you have to replicate your genome. And for a long time, we did not have the technical tools to study the genome of the non-cycling cell because there's just not enough DNA in there to study. But now there's methods to amplify and accurately sequence the genome of single neurons or single heart cells or single muscle cells. And the surprise, and somewhat disheartening surprise, has been that non-cycling cells, those that don't undergo cell division, accumulate mutations with age at a rate that's not much less than the rate at which dividing skin cells or dividing esophageal cells accumulate mutations with age. They have a particular pattern in which those mutations occur. They happen to be C to T and T to C mutations generally. And in fact, they resemble a signature that's long been seen in cancer. In fact, it is ubiquitous in virtually all cancers and is related to nothing other than the age of the patient at the time of diagnosis. And so this seems to be now, we can recognize a universal increase in mutations in the genome in cells with age, regardless of whether they're undergoing cell division or not. And the exact cause of this age-related mutational increase is not completely known. The best guess is that it might be something like Ben was suggesting, cytosine deamination. It might be a chemical process of the DNA. We've actually looked in patients who died with genetic disorders of nucleotide excision repair, and those single cells have more mutations than age-matched controls, but this particular aging signature doesn't seem to care whether nucleotide excision is working or not. It seems like it's something very universal. And so that would, I think, potentially put some limitations on the power of the sort of reprogramming, because we would still be reprogramming cells that have accumulated mutations. So this may be an aging clock which is not so easily reset as the epigenetic clock can be reset, or some of the other clocks can be reset. But it's also of vital importance to understand what exactly is this age-related mutational aggregate, accrual, and it's very, very linear with age.
I wanted to ask Don Ingber, are you still online?
Yeah.
I wonder, have you done aged cells in your organs on a chip? Don? Don?
I think he's probably getting a cup of coffee.
I think we lost you for a minute. Unmute. Can you talk now, Don? Okay, we'll come back to this question. Gilda, you had your hand raised. I actually don't see your hand raised. I'm sorry. Maybe it has to do with background issue. But anyway,
yes, I had a background issue too. But I like to express thanks too for being a part of this wonderful panel. I actually wanted to pick up on a point. I didn't want to lose sight of a point that Felipe made earlier. So we have this great basic science that's working so well that people are living so much longer. And then what do you do? Like, how do you address that? And I also wanted to, along with that, to pick up on a point that Eric had made about you really need a systems approach. So if we think about needing to collaborate across disciplines and bring together the basic scientists, clinicians, and engineers, those who are actually implementing, I wanted to give an example from engineering that has been useful to think about what do you do and serving the community long term. So we have this course that's called Engineering for Humanity, and it's specifically designed for aging. So bringing teams of engineers together, working directly with members of the senior community, so older adults, about what those issues are and how to actually design for that. So you take into consideration the equity component, like who has access to the technology. You get a sense of what are the needs, identifying the needs, and then coming up with ways to implement those solutions. So I just wanted to bring that up as an example of crossing these different areas and what do you do with the success of helping people who are living longer. I think it's something that we're gonna have to do more of, how we actually collaborate. Again, bringing the clinicians, the scientists, and the practitioners, and those who we are serving together So I'm hoping that piece would just add to the discussion a bit. So thanks for that.
Thank you, Gilda. I did ask a question about gender and issues of Susanna, you had a comment to make on that?
Yes, yeah, that was I wanted to follow up on that because actually it was not brought up yet and I was waiting, and I'm glad that someone raised the point of doing the basic animal research. We talk about sex dimorphism, that because the animal, they don't identify with a gender, but they have a sex. And this is a fundamental aspect that should be covered for anything that anyone is studying. Our favorite molecule, our favorite treatment. There are fundamental differences between male and female that are reflects both in the animal studies and in the human lifespan. I mean, we know, let alone the fact that there is evidence that women live longer, right? And so the mechanisms behind this living longer, what are the mechanisms and why? And so, and actually you mentioned something that is very personal, because when you said we want to go to Mars and we won't go there until we figure out the problem of aging, we are actually, our lab is really studying exactly that. One of the aspects we study exactly that problem of space radiation on the way to Mars. And to link back to this sex dimorphism, there is already evidence that if you put the animal on that stress environment that is in deep space radiation, female, for instance, respond completely different than male, and they are more resilient. And so, yeah, I don't want to take too much time, but that's a point where we have shown that, and many have shown that the immune system is respond differently, and this is especially in treatment, right? We know that many clinical trials have failed then on females because we know they function differently. So this should be heavily studies with any system that you want to work on.
Thank you very much, Suzanne. I just want to go back to Don Ingber. Can you hear us? I'm not able to hear you for some reason or other. That's too bad. Okay.
If I could jump in here real quick, just to say one thing to follow up on Susanna's topic of gender and sex differences with regards to longevity. One of the things that's also really exciting and important are aging of the immune system and differences in male and female, and immunosenescence, and the recognition that aging of the immune system and immunosenescence is going to be a very important component of both understanding underlying disease mechanisms and risk factors, as well as how we leverage the therapies, immunotherapy and other things for some of the brain diseases. So I'm really excited about understanding how we harness the power of the immune system to treat brain disorders and other disorders, not just how the autoimmune space has done it, but how cancer, cardiovascular disease has done it. But understanding how the immune system is aging is going to be key for us to understand how we use it better and more efficiently.
Felipe?
Yes, so this has been a great discussion, and I don't know where to start because, of course, with this Zoom system, you can't react immediately to when people say things. So just to finish on the male-female thing, I think it's something that the aging community is very, very aware of. In the interventions testing program, most of the drugs that we have tested have turned out positive, are either positive only on one sex or primarily in one sex. So it's something that the community is very aware of, and I think it's gonna transpose to when we do studies in the clinic. I think one thing that I would like to come back to is what Joshua was saying. So doing a clinical trial, having a frailty as an endpoint, I think it's very, very important development. Is precisely what Nir was mentioning as the problem with getting tame. Basically, the fact, will the FDA approve it? And the answer is yes, they approved it. And it's whether they will approve a drug being targeted to that, that's still not clear. But the trial was approved at least. And recently we did have approval of a drug against obesity. obesity is a risk factor for multiple diseases. It's not a disease on itself. So there is a precedent for the FDA approving things that are not really age-related diseases. So that's a point that I want to make. And also just to answer to Tachi's comment about, which is something that is true, that the research community also always suffers from the thing that, yeah, things have been fixed in mice, but not in humans. Yes, it's because many of the models that we use in mice are wrong. First of all, for example, you do a test in cancer. How old are your mice? Usually 2 to 3 months old. Well, that's not the same physiology as an old person who's going to get cancer. So it's not strange that it fails, because you're violating a central rule of science. You're moving 2 variables at the same time, the species and the age. So it's not rare, it's not strange that it doesn't work in humans. The other thing that I always make a point on this area is that because aging research, because we have been focused for so long on longevity, because it's expensive and time consuming, we don't test things in mice unless we already know that the intervention works in yeast, in C. elegans, in Drosophila, in fish, etc. In other words, we have the whole gamut of evolution and an intervention that works all from yeast to mice. And we're trying to move from mice to humans, which are right next to each other, really, compared to that scale. So that's the arguments I always use to justify, if you wish, that we do research in mice in aging research.
Satchin.
Yeah, thank you. I'd like to come back to this, combine 2 different things, lifestyle and also various drugs. One thing that is lacking now is how to define lifestyle. And then the way we are attempting to define lifestyle is what, when, and how much we eat, sleep, and exercise or move around on a daily basis. And the interesting thing is, if we now use this framework in clinical trials, then that will necessitate to collect these lifestyle factors and see how these lifestyle factors interact with the drug for intended outcome. So that might give us more insight into how to even take a drug that might have marginal effect and boost its effect by optimizing lifestyle. And this approach will also help us to address the health equity, because even if we take a very cheap drug that has a pleiotropic effect, and we can further boost its effect by optimum lifestyle, then that will have a wider impact. And coming back to Felipe, your comment about mouse and human, one big difference between mouse and human we always see from circadian point of view is mice are nocturnal. So most of the drug intervention that we do in mice during daytime, we wake them up from their sleep and we give them the drugs. And in humans, most of the drugs are administered during daytime. And acknowledging that difference even, and designing better animal housing where we invert the light dark cycle and work under red light might increase the chance of translating findings from mice to humans. And that's something that we have been doing in our research, and we are finding that there are many differences between what other people have published and what we find when we invert this. So I just wanted to combine these 2. And then the third thing that I wanted to bring up is we must not forget about diet. And in diet, what has happened is the last 200 years of agriculture has been random selection of palatable fruits, vegetables, and other things. Now the question is, can we bring molecular domestication to find active molecules, nutraceuticals, and other factors that can impact one or more, ideally pleiotropic effect on these 9 pathways of aging. And in that way, we can also address health equity, because if we can use molecular domestication to find new molecules that may be even widely available in certain crop plants, that will have a huge global impact on age-related diseases or reducing the burden of disease. Thank you.
Sachin, Tom?
So thank you. In the nonlinear way this conversation goes, I want to circle back also to Victor's question about reprogramming that I should have mentioned, and that is, what I highlighted was, I think, the risks of genetic reprogramming. But what I'm very excited about, and I think is really a kind of a forefront direction, is other kinds of reprogramming. And I'll specifically mention metabolic reprogramming. So the possibility of using metabolites really to modify the epigenome, and we know that's completely doable. And it links a lot of the biology of aging that we study in terms of metabolism with this idea of reprogramming the epigenome and maybe cellular age as being an intervention and an approach that takes advantage of the safety of metabolism and metabolites with this kind of promise of reprogramming. So again, I think I highlighted kind of the current riskiness of genetic interventions, but I think this idea of reprogramming cells in different ways has great promise outside of genetic interventions.
Great. Thank you. Andrew?
Yeah, hi. We're going to jump again, but actually, before I jump, following on from Tom's sort of idea and the risk of genetics, Tom's mentioned metabolites as a reprogramming. Recent work as a preprint in bioRxiv using transient reprogramming of 3 Yamanaka factors identified downstream targets in muscle, one of them being Wnt4. And so the other approach is find the direct targets. Now, it's likely to be organ specific. I'm sure Wnt4 plays, we know Wnt4 plays a great role in muscle, but does it play in other tissues or is it something else? And I think what's fascinating is that the reprogram seems to be consistently sort of having a boosting or regenerative effect. And one may imagine the downstream targets are distinct. So I think it's really interesting science there.
I really think so. And the reason I brought up earlier about the ethical issue is that you can push this technology, you can imagine, to do many things. And therefore it begins to ask the question, are you really trying to, in our traditional way treating disease, they're trying to enhance an organ, right? But I think it's a phenomenal technology.
Oh, I think so. I think it's captured everyone's imagination as well as parabiosis did, 15 years ago. So one of the technology things that I've been thinking about, I guess, is machine learning and the idea of complex biology and whether it's the age of someone's skin on their face or multiple biomarkers. And I think the advent of machine learning is going to have a big impact in how we view aging. The other aspect is wearable devices. And so I think this is where technology and health equity can sort of converge, which would be a device is much cheaper than going to the doctor and having prescriptions. But if we can own our own health span and we get feedback of what our blood glucose looks like, or our ketone bodies any day of the week. And so not only now are you seeing your own monitors, but you also know your own health history, right? You know what drugs you're on. And that brings into the issue of natural history experiments. So clinical trials are expensive, right? But we're all walking around with these devices. Now, of course, there's data privacy and who's going to analyze it. But maybe there are 10 million, 15 million people with a device on them right now. And so can we be looking at that data, whether it's exercise or diet? And Singapore, Victor, as you know, has sort of led the way in sort of helping mobilize the population into looking at their health span, their activity levels. But there's a whole bunch of people taking drugs, exercising, maybe having sleep disturbances. And maybe the data is there. It's obviously not a double-blinded trial, but it does allow different nations to get on to sort of think about health span and actually allows people to adopt the notion of aging being preventable or reversible rather than inevitable. I'll leave it there. Thank you.
Subject of machine learning, information technology, wearable tools, robotics, etc., will be discussed. We are focused here on basic science, biology, but the others are more applied science sessions, and will focus on some of the points you're making. So please join us if you care to, and you can benefit from the expertise in the room in those sessions. Other comments before I end the session with a summary, the best of my ability to summarize a broad-ranging discussion. I do request that we have not really focused a lot on emerging science directions. Those of you who have specific expertise in one area of science or another, who feel that that science is going to be critically important in our understanding of aging or solutions to aging, please submit them independent of this particular session. And I realize the Zoom format is very difficult because it's hard to have a free-flowing discussion if everybody's raising their hands and we're following a format that makes it difficult to have a continuous stream of discussion. So I welcome comments from any or all of you about ideas that you didn't have a chance to mention, or some thoughts you had on some of the discussion that took place.